Before we begin, a well-deserved acknowledgement. This video is based on a landmark study published in Nature by 𝗔𝗱𝗃 𝗔𝗱𝗃 and 𝗥𝗜𝗏, together with Christine Ziegler, Max Grammelspeicher, Lydia Fredolino, Zhonggang Ho, and Yan Zhang at the University of Michigan. Their meticulous work, years of phage assays, biochemistry, and deep sequencing, uncovered something nobody expected: a hidden second job for one of biology's most famous proteins. This is the story of how Cas9, the molecular scissors behind CRISPR gene editing, moonlights as a sensor that helps bacteria sense when their immune memory is running dangerously thin. To appreciate the discovery, start with the basics. Bacteria live under constant siege from viruses called bacteriophages. Their defense is CRISPR, a genetic scrapbook. When a bacterium survives an infection, it clips a snippet of the invading DNA, called a spacer, and files it into a CRISPR array between short repeated sequences. Later, that array is transcribed into CRISPR RNA, or ccRNA, which guides a Cas protein to recognize and destroy the same invader on site. In type II systems, that Cas protein is Cas9, and to cut DNA, Cas9 needs two RNA partners loaded together, the ccRNA, and a second RNA called tracRNA. Everyone assumed that Cas9 without its RNA partners, 𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏, was simply idle, dead weight. This study proves that assumption wrong. The researchers built their experiment around Neisseria meningitidis, the bacterium behind meningococcal disease, and its resident filamentous phage, MDA5. Neisseria carries a compact type IIc CRISPR, Cas9 system, smaller and simpler than the famous Streptococcus pyogenes Cas9 used in gene editing labs, and lacking extra helper proteins other systems rely on. By infecting Neisseria with the phage and sequencing the CRISPR array afterward, the team could catch bacteria in the act of acquiring brand new spacers against the virus, and directly ask what role Cas9 plays in the process. Cas9 already had one known job in acquisition: it reads the flanking DNA sequence, a short motif called a PAM, to make sure only functional, virus-matching spacers get filed away. But then the team deleted the tracRNA gene, and acquisition exploded. Efficiency jumped from a baseline of 6% to 61%. Deleting the CRISPR array itself, to eliminate ccRNA production entirely, produced the same explosive result. They called this runaway acquisition superadaptation, and it still correctly favored viral DNA over the host's own genome, and still required the PAM reading part of Cas9. Something about losing the RNA partners was unleashing Cas9's hidden potential. Removing both RNAs at once, no tracRNA, no ccRNA, produced the same superadaptation, and restoring either the RNA pair or a single fused guide RNA switched it back off. Biochemistry confirmed the switch directly. Purified Cas9 only formed a stable complex with ccRNA and tracRNA together, never with either RNA alone, and only the fully loaded complex protected Cas9 from enzymatic digestion. In other words, Cas9 exists in two distinct states: armed with its guide RNAs and ready to cut, or naked and RNA-free, as 𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏, and it's specifically the empty, 𝗏𝗏 state that supercharges spacer acquisition. So which part of Cas9 does the work? The researchers systematically deleted each structural domain and tested the results. Removing the nuclease lobe, the RUVC, HNH, and PAM reading regions, killed superadaptation entirely. But deleting the entire recognition lobe, the part that normally cradles the guide RNAs, left superadaptation fully intact, and now immune to being switched off by adding the RNAs back. The nuclease lobe drives acquisition. The recognition lobe is the regulatory switch that the RNA partners grab onto to keep that drive in check. Why would a bacterium want an RNA-free safety valve at all? Consider a CRISPR array being built from scratch, starting from a single lone repeat, the evolutionary seed of a brand new immune memory. The researchers recreated this scenario with arrays of increasing length. In the smallest arrays, cRNA and tracrRNA were barely detectable, and acquisition efficiency was highest, over 30%. As the array grew and RNA production caught up, acquisition efficiency fell in step, settling near 5% for full-length, mature arrays. A shrinking supply of RNA partners was steadily unmasking more and more 𝗥𝗜𝗏𝗃, and more 𝗥𝗜𝗏𝗃 meant faster spacer acquisition. This safety valve turns out to matter in real bacterial life, too. Sometimes a spacer in the array happens to target a beneficial piece of DNA, like one that carries antibiotic resistance. Under selection pressure, some bacteria escape that conflict by deleting a whole block of their own CRISPR array, sacrificing spacers just to survive. The researchers isolated these natural escapers and found exactly the predicted trade-off: shrunken arrays produced far less cRNA, and in response, their acquisition efficiency roughly tripled, from 5% up to 15%. Cas9 was sensing the cRNA shortfall and stepping in to rapidly rebuild the memory bank the cell had just sacrificed. Is this a Neisseria quirk, or something deeper? The team tested 7 additional type 2C Cas9 proteins from distantly related bacteria, sharing as little as 62% sequence identity with the original. In every case but one, the RNA-free form still triggered superadaptation, each accurately favoring its own species-specific PAM sequence, and adding back the matching guide RNA switched it off again, just as before. This isn't a coincidence found in one organism. It's a conserved biological principle, built into an entire branch of the CRISPR family tree. Put it all together, and a beautifully simple feedback loop emerges. A healthy, well-stocked CRISPR array produces abundant cRNA, which occupies nearly all the cell's Cas9, keeping acquisition modest and reducing the risk of the bacterium mistakenly targeting its own genome. But when the array is young, or shrinks, or gets damaged, cRNA levels drop, freeing up 𝗥𝗜𝗏𝗃 to sense that scarcity and aggressively drive new spacer acquisition, rebuilding immune memory before the next viral attack arrives. Cas9 isn't just an executioner. It's also a sensor, continuously reading the depth of its own immune archive and rebalancing acquisition in real time. For over a decade, Cas9 has been celebrated purely as a cutting tool, the scissors that made programmable gene editing possible. This study reveals a completely independent role, hiding in plain sight, 𝗥𝗜𝗏𝗃 as a self-replenishing guardian of bacterial immune memory. It reshapes how we understand tracrRNA and cRNA, not just as a guide for targeting, but as a built-in brake on runaway immune acquisition. And it opens real possibilities for biotechnology, from more controllable molecular recording systems to a deeper understanding of the CRISPR systems already deployed across medicine and agriculture. Once again, huge credit to Xiufei Zhou, Ruqing Diao, Xian Li, Lydia Fredolino, Zhonggang Ho, Yan Zhang, and their entire team at the University of Michigan, published in Nature, November 2025, for the patient, rigorous science that uncovered it. This research has also inspired the creation of a custom GPT, the Adaptive CRISPR Intelligence GPT. It is not just another CRISPR assistant. It is a full CRISPR research operating system, combining literature understanding, experimental design, evolutionary simulation, safety assessment, manuscript preparation, patent ideation, and commercialization support into a single expert assistant. By embedding this paper's newly discovered 𝗥𝗜𝗏𝗃 feedback mechanism as its core reasoning engine, it goes far beyond guide RNA design, positioning itself as a comprehensive AI platform for adaptive CRISPR research and innovation.